At the outset of our discussion about molecular clock, I'd like to bring in this diagram. This is a useful diagram when we're thinking about variation and diversity at the population level, what can happen as we get changes in that population splitting so that we get speciation events. And it's a lot to try and take in, and this is a hard connection for many people to make, but it's worth looking at to think about it. So in this diagram, the big fat phylogeny or tree represents the species relationships. But then within that, we have dots representing individual alleles in the population. And then when the dot changes, that means there's been a new mutation. And notice that some mutations might be under natural selection for a period of time. Others may be just evolving via genetic drift. That if there is a mutation in an ancestral population, it can be carried into both descendant populations. And this is a good one. If we did a phylogeny, if every one of these represents a new mutation or a number of new mutations to represent these new dots, we could very clearly define species. Despite diversity within a species A, B, and C, we can clearly define them. So we're not getting any lineage sorting at this level. We don't have any paralogy issues, so these don't get confused here. These don't represent two copies of the same gene, right? These are alleles, and that's an important distinction to make. An allele is a version of a gene. Everyone who is diploid, barring some strange mutation in human populations, everyone carries two distinct alleles for most of their genes. Now, if you're looking at a mitochondrial gene, it's a little different. If you're looking at an X chromosome and Y chromosome, those are a little different too because of the way that sex is determined in humans. But for the vast majority of our genes, you have two copies, okay? And so these alleles don't represent any one individual. They represent all of the alleles across a population. So at this point, you can think of it as 10% of the alleles, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. 10% of the alleles are one type of allele represented by this blue dot. 90% of the alleles are those gray ones. And as time goes on, we get more and more blue alleles until we never quite get complete fixation of the blue allele. And so at this point, we have quite a bit of genetic diversity. When the population splits, that genetic diversity is carried into both of the descendant populations, but notice we lose this light blue allele very rapidly, maybe even right before that species split. And then we get more patterns changing. So just be aware of that, okay? So the nature of the process that generates mutations is effectively random. There are some exceptions to that rule, and we're studying more and more about it, but by and large, generally, that's a good rule. So we'll stick to it. So particularly the point mutations that we're looking at appear when they first occur are random. Now, be aware that there are other things that could bias the outcome based on looking at just a data set of mutations in, say, a population or even across species. So even though the mutations themselves may be random, perhaps proofreading mechanisms are not random, right? So that might throw it off. Mutations themselves might be random, but then there might be some sort of a GC content bias, and so there's selection on certain types of mutation. And so even though the mutations themselves are random, the patterns that are generated, particularly over small regions, may not be completely random, because there might be some sort of natural selection or even sort of variational processes, mechanistic properties maybe is a better term, like biases in proofreading mechanisms, right? So we can think even of the mutation rate itself maybe on a couple levels. So when we think, what is the chance of fixing a new mutation in a population? Now, if natural selection is playing a role, this really is a very, very complicated question, and we're going to need to do a lot of work and a lot of research on that one allele, look at the other alleles, right? Compare it, the other mutations. But if it's not subject to natural selection, if it's truly neutral, then suddenly we've got a better way to do this. And in a lot of ways, trying to figure out the chance of fixing any one allele in a population, if it's completely neutral with no natural selection acting on any alleles, is a lot like predicting who's going to win the lottery, or predicting is a certain basketball player, is LeBron James going to make his next free throw, right? So there are, if we know something about the system, we can predict, even if it's a random system, we can make some really, really useful predictions, okay? So it turns out that the chance of fixing any one allele in a population, if it's completely neutral, is inversely proportional to the population size. Now let's look a little bit at this notation, okay? The N subscript E. In fact, I don't even know that that's a subscript. Let's make it a subscript because this is the most common way it's done. I think I've got to go font subscript. Okay, that's the most common way it's done. It's looked at. Okay, so don't get that confused, but that E is supposed to be subscript. So this is the effective population size. It's different than the census population size in this way. The census population size is every living individual in whatever population you are studying. Right now, the United States is going through a census count, and the idea is to try and count every individual. Now, in reality, they never are able to count everyone, but with a little bit of work, you can probably get a pretty good estimate of everybody in the population, and that would just be N. N subscript E is, in a population, the number of individuals who are going to reproduce and contribute to the next generation. And that's the number, the population that we are worried about with evolution. So for instance, if we count, I don't know, a turtle population, we count everyone, we're going to find millions and millions of turtles. Many of those are going to be quite young. They're going to be way more young turtles than there are old turtles. And only maybe 10% or less of those young turtles will make it to sexual maturity and be able to reproduce. So even though the Ridley sea turtle population might be 10 million individuals spread out across the oceans, its effective population size might be less than 1 million. I don't know what numbers are for Ridley sea turtle, but realize that in many species, in fact most species, effective population is much smaller than the census population. So the chance of fixing a new mutation is 1 over N to the E, and depending on whether it's diploid or haploid, we might have different numbers. You might see a 2 or a 4 out here, but the main idea is that as population, the effective population size gets larger, the chance of fixing any new mutation are that much smaller. So if we have a neutral mutation in a population of 100 individuals, essentially you have a 1 in 200 chance of fixing that allele because everyone carries 2 copies, so 100 individuals means 200 alleles. It's a 1 in 200 shot, and that's only if it's neutral, where all alleles have the exact same shot. So that's a pretty long shot, okay? But if it's in 1 in 1,000,000, right? It reminds me of, again, that clip. I think I showed it to most of the classes that will be listening to this lecture. That clip from Dumb and Dumber, where he says to the girl that he likes, what are the chances of a girl like you and a guy like me getting together? And she says, not very good. And he says, like 1 in 100? And she goes, no, like 1 in 1,000,000. And the joke there is that you expect him to just be devastated, but he's excited because at least there's a chance. But if I propose that to you, if I said, hey, you've got a 1 in 1,000,000 chance, are you going to bet money on it? Or are you going to invest resources into it? You know, probably not. You're like, oh, that's probably no chance. But it turns out that humans are not so good at understanding math sometimes. So I look at you and say, hey, you have a 1 in 300 million chance of winning the lottery. And you're like, oh, cool, sign me up. I'm going to buy tickets. When in reality, you should be running away saying, oh, that's nearly impossible. Okay? But the same thing applies. If we have a brand new mutation in a large population, it's very unlikely that it will be fixed, meaning become the most predominant, the only allele in the population. So make sure you understand this term fixing or fixation. It doesn't mean repaired. It doesn't mean getting better. It means going from a state of being one of many alleles to a state where that one allele we're looking at is the only allele in the population. Okay, so fairly simple math. I tried to explain the reasoning why behind it. You could see it here. If we've got only 10% of the alleles are that, then we would have a 1 in 10, a 10% chance of that blue allele becoming fixed in the population over time. And then, of course, we might have new mutations that pop up that then complicate it further. Okay, so if we look at the chance of a new mutation, we have this fairly simple equation here. So you're already familiar, again, just to be clear, I guess when I transfer this over, I don't think all my subscripts came through. So let's make sure that, because this is the standard notation, let's make sure that all of them are subscripts here, all the E's. And I don't know if it will let me do repeat. Oh, no, it didn't like that. So I'll just do it the old-fashioned way. Okay. So there we are. So let's take a look at these next parts of the equation. And this is a very cool prediction of neutral theory. So we already looked at the chance of a mutation becoming fixed in a population. We looked at the chance of a new mutation occurring here, right, which is proportional to the population size, effective population size, and this little symbol mu, which represents the mutation rate. And again, if you're a diploid organism, we have to double that because you're getting one from mom and one from dad. You got 2 copies. So 2 times the mutation rate times the effective population size is the chance of a new mutation. And that makes sense. If you have a little tiny population and you have a certain base mutation rate, like maybe only 1 out of every 100 billion times you copy a piece of DNA, do you make a mistake? That's a very, very low mutation rate. Usually they're a little higher than that. But every time you copy your DNA, there's a small chance of having a mutation. And so if you have a very large population, you're copying your genomes more often, you're going to end up with more mutations occurring. But here's the cool part. These 2 forces offset each other. So the chance of getting a new mutation and then the chance of getting it fixed in the population, so you have more mutations in large populations, but there's a much lower chance, a proportionally lower chance of having that mutation become fixed. In a small population, you have a much lower chance, in a small population, there's a lower chance of having a mutation, but a much higher chance of having it fixed, and it's proportional. So in reality, when we combine the mutation rate and the fixation rate, and this is sometimes called the overall substitution rate, so the rate at which one base pair gets swapped out for another one is simply proportional to whatever the mutation rate is. So, and again, remember, this is only for neutrally evolving things. So if we can estimate the mutation rate, then we know how often we would expect to have one base pair substituted out for another base pair. And so if we have a population of a certain size and we look for how much differences there are there, and we know the mutation rate, we know how long that population has gone from descending from a common ancestor. So let's add in those terms here just so you're aware of them. You can put them in your notes. Right? This was also sometimes called the substitution rate. Make that smaller so it all fits in. Okay, so that mutation rate becomes a stand-in for the substitution rate. And then we can use that substitution rate to help us make predictions about divergence time. Okay, that's also called coalescence time. When did all of the diversity we see in a population, when did it last have a single common ancestor? Okay, now that's really tricky in sexually reproducing populations because of genetic recombination. But in bacteria, because there's no genetic recombination, it's easier. Horizontal gene transfer flow throws a little wrinkle in. But between species, it actually becomes a very powerful tool because in eukaryotic species, we don't really have mixing after true speciation occurs. Sometimes there's this gray area where species can still reproduce for short periods of time. But for instance, we don't have the diversity in humans being mixed with the diversity in whales, or the diversity in mice being mixed with the diversity in rabbits or plants or any other species. So once they're completely separated, truly speciated, then suddenly all the diversity that builds up in that population, is, if it's being governed by neutral evolution, is now only going to be governed by the base mutation rate. Now, not all regions of the genome are going to reflect the molecular clock, particularly genes that are under selection. And we can even see different levels of selection, right? The number of changes are indicative of the amount of constraint on that gene. So this is something that's in the Ney paper, if you read the paper for today or for last week, and also in the Bronham paper talks, I think, a little bit about this. But this shows a level of constraint and that there's different forces of natural selection acting on these genes. Fibrinopeptides are much more able to mutate and evolve and change over time. Cytochrome C, a very important conserved ancient metabolic gene, has very low rates of substitution even at the neutral sites. Okay, even there might be difference between the third base pair or the third position in a codon versus the first and second position in codons. Okay, but this shows that these would probably not be subject to a molecular clock because there's different evolutionary forces acting on them. And when natural selection is acting on a protein, especially if that natural selection varies over time, we cannot invoke and use a molecular clock. Now, one other aspect before we talk about some of the things that we can do with the molecular clock. The molecular clock needs to be calibrated, right? If we are able to estimate mutation rate. We don't know if that's always been the same in the history of these organisms. And so a very important part of estimating the mutation rate is calibrating the molecular clock. And so to calibrate the molecular clock, we can do it through a fossil record, or we can do it now, if we have large enough data sets and large enough sample sizes, we can look at levels of divergence over short periods of time, levels of divergence over long periods of time, and see if they've changed, and then maybe help to estimate, okay, maybe we've got a flexible molecular clock that's changing a little bit over time, and that would allow us to calibrate it within a certain range. Fossils allow us to calibrate the molecular clock too. If I find a fossil, I know it's the common ancestors of all amphibians, right, which I think in this image is this guy, right? If I find a fossil that represents that and I can date that fossil, then suddenly I have a set period of time over which all of the diversity in that group must have been generated. And it's a minimum amount of time, because when we find a fossil, that's a minimum date. There could be fossils earlier that we have just missed. So finding fossils that represent common ancestors allows us to calibrate the molecular clock. And that, in fact, is the most common way to do it, with some methods that we're not going to get into, some complex methods that allow us to estimate it over periods of time, even if we don't have a fossil. But fossils serve as anchors to calibrate the molecular clock.